So this OmicsLogic Metagenomics program is focused on bioinformatics for the study of microbiome using next generation sequencing. This session is a first session. We'll do an overview of the program, give you some idea of what topics will be covered in interactive sessions like this one today. We'll also discuss the different other resources that you will have access to during this program. That includes some online course material that you can complete on your own time, as well as coding exercises and some practical exercises using the big data analysis platform, T-BioInfo. But before we jump into that, let me tell you a little bit more about these kinds of OmicsLogic programs and about our team. So the OmicsLogic programs are a result of an amazing effort and collaboration between Pine Biotech, which is an innovative big data analytics company based in the US, as well as numerous institutions that have participated in content evaluation, curriculum adaptation, and project proposals. As a result, the OmicsLogic programs have been thoroughly tested and are proven to lead to excellent outcomes for both students and faculty that are interested in bioinformatics or the application of data science to biological and biomedical research. For those that are joining us for the first time, my name is Ilya Brodsky. I'm the co-founder and CEO of Pine Biotech. Our company vision is to enhance human health and well-being by enabling biological research and discovery with relevant data, big data analytic solutions, and support. Our mission is to simplify bioinformatics and advance research through our modular and intuitive multi-omics analysis platform powered by human experience and artificial intelligence. We offer training, research support, and big data analytics tools designed for research and biomedical discovery. And our mission is to support cutting-edge research projects and solve big data challenges by making our solutions easy to use and to provide training for those interested to use them in an effective way to arrive at sound and reproducible results. I am honored to have been working with a large team of people that have much more experience in this area than myself, including some of the other co-founders, Dr. Alfred Tauber and Dr. Leonid Brodsky. Together, they have designed and developed an innovative system that allows us to conduct such programs and is supported by a team of experts in bioinformatics, computer science, and other areas that help us form, organize, and run programs like these. So OmicsLogic Metagenomics is a program that is starting today on August 31st, and it is designed to be equally rewarding to both students and researchers. It does not require any background in bioinformatics itself, and it is especially suited for project-based learning on a flexible schedule. The program will cover main aspects of metagenomic sequencing, analysis of 16S ribosomal RNA, and other aspects of microbiology research utilizing bioinformatics tools and databases. At the end of this program, participants will be conducting independent or group projects, analyzing data from the public domain, and learning from high-impact biomedical publications on the microbiome. So to introduce the program, the resources, materials, and the schedule, we'll take a look at what microbiome is and how you will be able to learn about bioinformatics for microbiome research in the next few weeks. So since the discovery of the microscope, humans have been learning about microorganisms that surround us. And as the resolution improved, more and more representatives of the unseen world have been discovered. The scale of optical resolution can reveal details of up to 1 micrometer, which is a typical size of bacteria. Today, microscopes are tremendously powerful, allowing humans to see smaller and smaller elements that reveal how the world of microorganisms functions on every imaginable surface and is actively interacting with each other. In this image, you can see a super high resolution image achieved with helium-ion technology that shows the process of bacteriophages in green attack the E. coli bacteria. So the E. coli is blue and the green are the bacteriophages. The connection between disease and microorganisms has been established in the 1800s, and already towards the end of the 19th century, laboratory methods for growing microorganisms in a lab culture were developed, helping characterize them with staining. These studies provided important information about bacterial function in isolation. However, it was soon apparent that most microorganisms live in complex communities. Today, we know that the microorganisms, or the microbiota, that make up the human microbiome are not just unicellular organisms living alongside each other, but instead form a highly regulated, structurally and functionally organized communities attached to surfaces as biofilms with interspecies collaborations, as well as antagonisms that contribute to ecological stability of these communities. The stability of these communities is fascinating, as many of us probably know from trying to battle such common skin conditions as dryness, eczema, or acne. They form distinct communities, and these microorganisms are interconnected and determined to maintain their community. Bacteria within a biofilm can communicate with each other by producing, detecting, and responding to small diffusible signal molecules in a process called quorum sensing, which confirms benefit for host colonization, biofilm formation, defense against competitors, and adaptation to changes in the microenvironment. Quorum sensing activities in biofilms are also involved in the virulence and pathogenic potential of bacteria, and are therefore an important factor in understanding and controlling bacterial infections, as they enable microorganisms in biofilms to become more tolerant or host defense and antimicrobial agents. Inside each community, you will find that there is a variety of compositions and different types of bacteria that are dominant, providing some insight into their functionality, but also giving us a better understanding of these community changes as we study different types of conditions. The human microbiota is estimated to be 10 to the 13th to 10 to the 14th of microbial cells, with around 1:1 microbial cells to human cells ratio, even though it was previously reported that there's a much greater number of microorganisms. These figures have been updated by Sander et al. In 2016. These numbers are derived from the total bacterial cells in the colon, the organ that harbors the densest number of microbes. However, each one of these microorganisms carries important functional properties in genes that contain genomic information and can give insight into their functionality. These properties and the genomic information about microorganisms have been of a growing interest to the scientific community, resulting in a total over 80,000 publications. The majority of these studies have been focused on the intestinal microbiome. But since the 1990s, microbiome studies were significantly advanced through such major initiatives as the Metagenomics of the human intestinal tract and the subsequent Human Microbiome Project. These are very large studies that have been made possible through the advances in metagenomic sequencing. As a result, skin, lung, gut, and even brain microbiota is starting to be characterized and better understood to reveal important relationships between the microbiome composition, different types of species of microbes, and conditions, and their onset in the humans. So many of these questions can be answered using two types of technologies that we will review in this program. Primarily, we will be focused on next-generation sequencing. Next-generation sequencing allows us to study the composition or specific genomes and genes that are being expressed by microorganisms. It can also be studied using mass spectrometry. Mass spectrometry is a technology that allows us to capture portions or whole proteins and metabolites, allowing us to understand what are the specific products of gene expression and signaling between these bacterial organisms, between these microorganisms. Our primary study in this program is going to be on metagenomics. So metagenomics is different from other types of omics data because genomics, proteomics, and transcriptomics all focus on the whole of a single organism. So for example, we want to study all of the genes or proteins that are expressed in a given organism. Or we want to understand variation of DNA across cells of an individual. But metagenomics is the study of the composition or the whole community of particular microorganisms. So it allows us to study these communities not individually, but as a part of this larger community. I hope that in this program, we will be able to learn about the microbiome and its function, which includes differentiation of the immune system, food digestion and nutrition, energy generation and conservation, metabolic regulation, processing of environmental chemicals, maintenance of skin and mucosal barrier function, and prevention of invasion by pathogens. So it really does affect a lot of areas of human health, preclinical and clinical conditions, as well as treatment. Oh, sorry. Okay, so in this program, we will utilize several different resources that I want to review in this session. The first one is we will have a series of interactive sessions, just like today. We'll get to the interactive part in just a little bit. But I want to make sure that all of you have received the invitation to this session. Obviously, you have if you're on the session. But you will also receive regular emails from marketing@pine.bio. So it's important to add that email to your address list, to your contact list, so that you will receive these invitations regularly. And we will do this on a predetermined schedule. And afterwards, when we will have project review sessions, we'll also send you custom invitations for that as well. The second part that we will review today are some asynchronous courses that are found on our learning platform. So we will review that in just a second. In many of these courses, you will see assignments, and these assignments leverage our analytical platform, as well as some additional resources that we will cover today as well. So you should have received with the email about this session, your passwords for the access to these different platforms. So in the next few slides, we'll review how to access and utilize those in this program. Okay, so let's first make sure that you have the login information for server.t-bio.info and edu.t-bio.info. How do you know that you have access? So the first thing that you can go to is this page. Once you get to this page, please let me know that you're there by answering yes in the chat box. Or you can write, yes, I'm there, got it, something, so I know that you've arrived at this page and you see what I see. Okay, did anyone get here? All right, great, Shreya is here. All right, great. So those of you that are registered in the program will have access to everything that I see. Some of you already know from some of our previous programs, so I assume you already know most of what we are going to discuss today in terms of access. All right, great. So a few important places that you will find, these are specific resources for this program. You will have a forum where we will post information about upcoming sessions, recording from the previous session. We'll have forms, as well as different links for additional resources. On this tab, you will find courses related to this program. We'll have some project examples. We'll be keeping progress record for all of the participants of the program. So we'll review some of this progress and make sure that all of the challenges in terms of technical challenges are being addressed. And also you will find here under menu, the syllabus tab. So if you go to the syllabus tab, you will see an overview of all of the sessions that we will cover and some associated resources for each one of our sessions. So please make sure that you can navigate to all of these links and you can see the information. If you have not yet completed your registration, you can also complete your registration. And if you have any trouble completing the registration, you can reach out to marketing@pine.bio and we'll help make sure that you have the right links and can complete your registration. The first thing that you can do is you can start with the course called Introduction to Bioinformatics. Introduction to Bioinformatics is not related directly to metagenomics, but it also helps us to get to learn about you. So this program is to learn about microbiome and metagenomic sequencing, but also helps us if we understand your background and your interests, so we can best adapt our material throughout the sessions to make it most effective for you. So if you go to this Introduction to Bioinformatics course, this is a free course, so anyone can actually take it. One of the first lectures that you will see is Intro Survey. So in the survey, if you fill it out, we will receive this information and it will help us make sure that information is available on all of the participants and we can make the most of this program together. Okay, so for this particular survey and this course, everyone should be able to have access. It's free. You do not need to be a participant of this program specifically. Okay, so please take a moment and fill out the survey. If you have filled out the survey previously, you should see your information already here. After you have completed this survey and went through this course, which I think is going to be informative, even if you have a little bit of background in bioinformatics already, you will also understand how the system works. And then there are several courses, and I've seen that some of you have already started taking some of the courses, which is great, which are Introduction to Metagenomics, Metagenomics 1, and Metagenomics 2. So in these courses, we'll review some of the same topics that we will cover in the interactive sessions, but we will add additional depth and additional project examples in the interactive sessions that we will have. Each one of the courses is organized in the following way. You have here expectations, so it's important to understand the terminology and the biological context, as well as different types of data formats and analysis logic that we will explore during this whole session or this program. We'll try to utilize some aspects of topic mapping so that it's clear why are we applying specific data analysis methods to a particular type of data, and what are the specific outcomes of that analysis that we're interested to take away to be able to interpret some biological phenomena based on our analysis results. You will also see a number of different tutorials and multimedia elements that help explain different types of data sets and projects. These are going to require some additional reading in many cases so that you understand the project as a whole and have significant background in that project before we start the data analysis part. In the analysis section, we will utilize some practical analytical steps. And these practical analytical steps are organized in some kind of a logic. So once you have understood the logic, we will also look at some practical assignments that will, if you are interested in going that far, utilize some coding elements in R and Python. So we will discuss some exercises that will help you make the most and be able to improve some of the visualization outputs and also ask some specific questions from the data set that we do the analysis for. And so to keep track of all of these kinds of activities, we have developed a number of tools that help both us, but also for you, understand your progress as compared to the rest of the group and spot any challenges that you might be facing. So for example, we can see when you run a particular pipeline. We can also take a look at the outputs of that pipeline to make sure that you've gotten results and address any issues that come up. There are different coding exercises that we will review together, and to make sure that everything is set up, since we don't have access to your computer, we have created this online interactive platform that allows us to also see that you have completed everything without the need of installing different libraries and resolving any kinds of technical issues that typically arise during installation, and we don't have access to monitor that. So in the online courses, specifically for metagenomics, you will see that we cover a number of different pipelines. Pipelines are connections between different executable scripts and algorithms that process data in a step-by-step manner. So here you can see an example of one of the lessons where the data to bioinformatics workflow is reviewed. So we'll do a number of review sessions on these types of pipelines, discuss the specific steps, why these steps are necessary, and what types of outputs do they produce so that you can continue and explore the data in a more detailed way. Okay, so since I've seen that some of you have already done some of the preliminary coursework, I wanted to ask a few questions, and that will also give us some sense of where you are. So it's okay if you don't know some of these answers, but if you do know some of these answers, you can put your answers in the chat box after I ask each question, and then we can just briefly review what the answers are, and we'll continue to do such reviews in our future sessions. So the first question is, which of the following terms is unrelated to 16S ribosomal RNA? Okay, so here we have a few people answering protoplasm. All right, excellent. So many of you already know the answer. Let's review why that is protoplasm. So can someone tell us what is protoplasm? Okay, so Deb is saying cytoplasm and organelles. So what is protoplasm? Okay, so it's the cytoplasm equivalent of plant cells. Excellent. So protoplasm is the complex semi-fluid translucent substance that constitutes the interior matter of a living cell and is composed of proteins, fats, and other molecules suspended in water. It includes the cytoplasm, the nucleus in eukaryotes, and organelles such as mitochondria. So this does not have anything to do with 16S ribosomal RNA. Why? Because 16S ribosomal RNA is the gene of the ribosome that could be used to study microbial taxonomy. And the other terms in the answer here, amplicon, is sequencing that amplifies 16S ribosomal RNA, or one of the hypervariable regions. Next generation sequencing is what we use to study the 16S ribosomal RNA gene. Phylogeny is our representation of taxonomy that we can look at for different compositions of the microbial communities. And hypervariable is a term of a particular region in the 16S ribosomal RNA gene that allow us to understand the taxonomic composition of a particular community. Okay, so this is a question that helps us study some of the terminology. Okay, so what is taxonomic classification? Taxonomic classification can best be described as 1, studies of increased rates of disease within a collection of microorganisms. 2, new ways of simultaneously sequencing multiple genomes. 3, studying the extent to which genetic sequences diverge. 4, a way of preparing genomic material to be sequenced, or 5, the removal of variable regions of RNA to increase experimental validity. Okay, I'll just give a few seconds here. All right, excellent. So all of you are familiar with taxonomy. It is studying the extent to which genetic sequences diverge, which allows us to understand how they form different groups. Okay, and finally, shotgun sequencing is an important technique in metagenomic studies. Which of the following are true regarding this type of sequencing? Number 1, shotgun sequencing is based on multiple reads. 2, utilizes a reference genome that is compared to the sequence reads. 3, can be performed on DNA and RNA. Or 4, all of the above. All right, excellent. So all of you, I see, have a lot of background information already on metagenomics. Taxonomic classification is a way to study metagenomic communities, and we use shotgun sequencing, so it's important to understand some terminology related to what this technique actually does. So it's really good to see that the majority of you have some background in these topics. For those of you that do not, I recommend taking the Introduction to Metagenomics course, and that will give you a lot of information about all of this terminology. And for those of you that know but need a refresher, that's also a good way to evaluate how much you remember. So to analyze this kind of data, as I mentioned, initially you do not have to have any background in coding. In fact, we will use a point-and-click interface to understand the logic of analysis, and this logic of analysis will clarify why we are taking certain steps and what is the objective for each input and output in this process. So these are going to be called bioinformatics pipelines. For those of you that are already familiar with bioinformatics pipelines, we will actually take a look at some of the settings and the options that allow us to study the data and extract different types of outputs from this data. We will also then later on go into the specific code that allows us to run these different steps and see what kind of variation we can work with using coding. So let's make sure that you have access to this platform. So the link is server.t-bio.info. All right, please let me know once you are here. Okay, so the link, this is the link. All right, great. So for those of you that are here, first of all, if you are logged in, you should see your profile details right here. For those of you that have registered for the program already, you should have gotten an email with your access details. So you will have your name and your password in the email. There are several different levels for those of you that are interested in using this platform. There's a demo level, and this is the fastest option, right? And I want to stress that. If you want to just understand how different types of methods are used and what are these bioinformatics pipelines, you do not have to run any kind of analysis. So for example, you can go into this demo, read about a particular section, and then just run a simple pipeline that will not take up any of the server resources and provide you with an output that you can still discuss in the context of a course. So for example, I have here a completed pipeline and several outputs that I can click on to download and explore. You can get this kind of access for a demo for free. So even those of you that are not a part of the program, but you would like to understand how these different types of pipelines work, you can request free access and you will be able to run these kinds of demos. To request that kind of access, you click right here and you will see a form. This form will be sent over to us and we will email you the details. The second level is an educational level. So for education, you can run any kind of public domain data sets. We will review this in the context of the program, but it's important to understand that either this data set is going to be provided to you, so specific links to data sets are going to be sent over to you with an explanation of what to do. You can find those in the lessons, in the coursework that we have discussed, or you can find them from public domain data sets like NCBI. So on NCBI, you can search and find specific data sets, and then we will tell you how to use those data sets in the analysis. The final level is research. When you have access to a research account, you will have an FTP account where you can upload data sets of your own from your lab or from wherever you have those data sets stored, and actually reference those data sets on this big data cloud-based platform. What's important to remember is that there are several places where you will constantly kind of refer to and look. For example, you can see previously completed pipelines that you can always go back to and look at what the output was and how you got to that output. You will also see a report on each one of your pipelines right here. Also, you will go back and you can go into all of the different sections. And for understanding of what these sections do, you can actually hover over these icons to make sure that you know what the section does. Inside a particular section, as I mentioned, once you are using the educational version, you will have to select some inputs. And in those inputs, you will determine what types of pipelines are possible. Okay, and this is a great time for questions. So if you have any questions related to what we've discussed up until now, please put them in the chat. For example, Shreya is asking, if I don't have a data set from research, how can I get an experience of data analysis? So as I mentioned, inside the coursework, you will get some data sets. They will be given to you. You can also, and we will discuss this even today, you can also start looking for data sets on public repositories like the National Center for Biotechnology Information. So for example, here, and we'll discuss what types of data sets are going to be available for what. If you click on microbiome, you will see a variety of different data sets available for free. These are publicly accessible databases that you can get experience of data analysis with. Does that answer your question, Shreya? Great. Yes, and for Prerna, I will definitely show the results of a pipeline and how to interpret and understand the outputs. That will be the topic of our next session. In our next session, we will start looking at what types of next generation sequencing data sets there are, what types of pipelines you run to analyze them, and what would be the outputs. And obviously for outputs, there will be a variety of different types of outputs, and we will discuss how to interpret the different types of outputs. Okay, Prerna, does that answer your question? Okay, and Asmita, yes, we will definitely, we have suggestions for books. Today we will review 3 different articles that I think you can start with. One of them is a chapter from a book. And that chapter talks about microbiome research and some typical methods of analysis that you have to learn about. So we'll do that at the end of today's session. To get started, I recommend some of the coursework that I already mentioned, where you can both read about, practice, and evaluate your understanding using quizzes and assignments. Okay, thank you, Deepak. If anyone else has any questions about this, I'm happy to answer about the resources that we've covered, but I have a few more that I want to get to towards the next part of this presentation. So again, just a brief overview of this platform. You will find multiple sections for sequence analysis. For example, we will be relying on next generation sequencing data to study metagenomics. You will see that inside you have some demo pipelines that you can click on this drop down and see the inputs, outputs, and what the steps are to analyze specific types of data. In our future sections, we'll talk about data mining and how to use machine learning and some utilities that will be useful for this type of data. And you'll also be able to go back and see all of your running and past pipelines. So you don't have to open a pipeline and wait for it to finish. You can leave. You will then get a notification that your pipeline finished and return to see what are the outputs that you are getting. And obviously, to check that you have your profile and you're logged in, you can always go to your profile. Okay, so another question here from Sultan Mohideen. Metagenomics is applied to all organisms. If I want to study on some organism and the data is not available on NCBI, then how to proceed with the research? Unfortunately, if you don't have data, it's hard to study something, not being able to generate that data set yourself. So we will try to focus on specifically microbial or microbiome research where we can find publicly available data sets. If you're interested in other topics and there are no data sets available on that topic, you'll either have to generate that data set or potentially that research is not possible. Yes, so definitely you have to generate the data if you are interested to apply bioinformatics to study that data. Okay, so what are some of the sections that we will review? The first section that we'll talk about is the metagenomics microbiome section that allows us to map reads onto reference databases, look at outputs that tell us what are the specific operational taxonomic units, or even full 16S ribosomal RNA genes that are present in a database. Then we will turn to analysis of 16S amplicon data, where specific hypervariable regions are amplified, and then standard tools for analysis of composition, species richness, and diversity can be applied to the data set and visualized using different types of graphs and charts. To practice some of these methods, we will also use some coding and analysis overview using the code.omicslogic.com platform. Okay, so let me just show you an example. If you go to code.omicslogic.com, okay, you will get access to different courses sequentially. You have to log in using the same email address that you have used to register for the program. So first of all, you will get, you will not see what I am seeing. You will see a landing page and you have to register or log in. So when you do that, use the same email address. Once you log in, you will see some of these types of tutorials. So please let me know once you see what I see on the screen. And okay, let me answer the questions. In the meantime, please, yes, tell me yes once you've gotten to this page. For Adarsh, what are other specific databases for microbiome alone? So there are some 16S ribosomal RNA gene reference databases like Silva, Green Gene, and others, RDP. There are also different projects that we will review today. For example, the Human Microbiome Project. So we'll take a look at those today in this session. Okay, so Adarsh and Mohammed, let me know if that answers your question. And Mohammed, your other question, how can you get the course for free? You're welcome to join the Introduction to Metagenomics is a free course, so you're welcome to enroll in that course. Okay, did anyone else get to this page? I'm gonna wait until, okay, great. At least 5 people get access to here. So we have, all right, excellent. So here you will see some basic introductory materials that you can already take a look at. For each one, you will see that there's an exercise in both R and Python. So you can already start exploring how to do some of these basic things. What we will do, okay, so what we will do here for those of you that are registered is we will review such packages as microbiome, phyloseq, including some methods of analysis like NMDS, PCOA, and SVD. So those of you that are registered for the program, we will do a little bit of coding, and this is only if you're interested in. So you don't actually have to do programming if you don't want to. If you want to, you have some options here. We will mostly rely on R for this program. For other programs, we do, Python is also useful as well. But in this case, we will primarily work on R. Okay, and for those of you that need some introduction, here's some introduction to data science. Again, specifically, yes, and I will explain the points here in a few minutes. So specifically, the packages that you would want to install are microbiome, phyloseq, and you can also explore KIME and also Mother. How can you find this page? It is code.omicslogic.com. And then for those of you that are just starting, you can go to About, and here you will find some guides on using this system. So this is essentially an R server. You can see that the analysis works on the server and produces results that you can take the results with. You will have some explanation and then try some things yourself by changing some of the code and running that code. For example, I can copy this link, change this link right here, press on run, and you can see success running code, and it shows me what I got. So there's actually no installations that are needed. And for Gregory, yes, I will get to that in our future sessions, but today I'm just doing an overview of the different types of things that we are going to review. But we will review both KIME and Mother. Okay, so take a look and play around with this. After you complete this whole section, you will start getting points. Okay, so what are points? We have a particular syllabus. This syllabus includes completion of courses. For example, here's a course. Once you go through all of the lessons of the course, sometimes quizzes and assignments, you have to press on Finish Course. And once that course is finished, you will see that you get a certificate for each course. Let me give you an example. So here's a participant from another program that we have. You can see her badge. Yes, you can. Yeah, you don't have to read through everything, but you have to submit all of the assignments and quizzes to get the results. So here you can see this person has almost 20,000 points, and these 20,000 points have been a result of this kind of activity. For example, she completed coding steps, she ran some pipelines on the server platform, she completed some coursework, and you can see over here under courses, and this is true for all of your different profiles, you will see how once your course is completed, it will say completed right here. So you'll see all of this. You also will see certificates. So for each one of your courses, you get a certificate of completion for an individual course. Once you have completed that course, it looks for both points, assignments, and quizzes to generate this kind of certificate. Now, yeah, so you can see all of the different certificates. You also get a certificate for the program itself. The goal of this type of a profile is to both track progress, address any challenges and questions, but also for you to take away and build up your profile. You can look at some of the top profiles over here. Okay, and I will pause again for questions to address all of your questions. So please continue to post your questions on the chat. So once I get there, you will see that we will address all of those. So you can see here some of the top profiles. You can learn about them, and we will also hope to see all of you in this list. So you can see that it goes for quite a long time, and you have to reach at least 11,000 points to get to this list. Okay, so what are some of the things that we will talk about? We have some basic courses, and you will see also that there will be tracking of progress as you make your way through these courses. And specifically, we will look at how to study and visualize results of the big data analysis pipelines, how to study composition of your samples, and look at some of the popular packages that typically are used in microbiome studies. So these are some of the things that we will cover, and most of them, as you can see on the screen, are going to be in R. Okay, so let me address some of these questions. So what are the specific databases? I think we answered that. You need this. So can we talk about OTU tables and how do we? Yes, so we will talk about that. Yes, definitely. Do you need to download them? Again, yes, if you want to continue and use this for your research, obviously it's going to be useful for you to have that package already installed. There's some guidance on how to install R and Python. If you want to, we will send you the link. Which OS will be better? Any OS would work, Windows, Mac, Linux, whichever one you prefer is fine. There are some challenges associated with each one. So we will try to help you address any of those, but we also provide the cloud platform so that we can avoid getting stuck on the specific installations and understand what we're trying to do. Okay, how to make SVLs through FTP. For Sana, we will send you a video on how to do that. And then if you have any challenges with that, we can schedule a session to discuss. Okay, so let's review how are we going to cover all of this material in this program, because we're obviously not going to have all of the time in each one session to cover all of these different aspects of data analysis. Yes, and all of these sessions are going to be recorded and sent to all of the participants of the program. For this specific session, this is a free session, so we will be posting it on YouTube. And if you follow our YouTube channel, Beeps, and maybe you could put a link to our YouTube channel, that channel will be where we will have this video. Okay, so you can see an overview of all of the different sections, or all of the different sessions, sorry, that we will have on this link. Okay, I'm just going to show you where this link is to the syllabus. Okay, great. So here you can see what we will cover in each session. For example, here we are doing an overview of the entire course, the tools and resources for the program, what are some of the expectations and schedule, and some of the important deadlines. So if you scroll through here, you can see that we are starting on August 31st, and then we will go through September 2nd, 4th, 7th, 9th, 11th, and 14th. And so that will be the end of our theoretical part of the program. The objective is to get you to start thinking about a project proposal or a research question and where to find the right data. So for Mohammed and Adarsh and others that have been asking what are the specific data sets and how to do that kind of analysis, that is where we will get to by September 14th. It will take us some time, but we will review types of sequencing, types of data, types of analysis pipelines, what are some example projects, and then we will start looking for those projects and also think, how do you actually plan for and structure your analysis so that the project that you have developed could both be completed in a short period of time, you understand the significance and can communicate the significance of this project to other participants of this program. So your goal is to do this by September 14th. So you have some time, but we will review. You can already start thinking about it, and I will give you some guidance today on how to get started with that as well. Okay, and I see that there are a lot of questions. So are there any other questions about what we discussed so far? So again, yes, we will work on an independent project. The deadlines, September 14th is when you have to submit your project proposal. Up until then, you can work on that project proposal. There's some course material that you have to complete. There is a certain number of points, and that's the last point I'll make about how the points work. You actually have to get to either an independent project completion or 10,000 points to get a certificate of excellence. For all the other participants that have done at least a minimum of the basic courses, which are 3 courses, Introduction to Metagenomics, Metagenomics 1, and Metagenomics 2, those will get a certificate of participation for the program. Are there any other questions? How long is the project proposal? We will actually review what the project proposal is. It is not very long. It is similar to the objective of the project is to be similar to a poster presentation at the end of the program. But when you say how long, I think that it's only a few paragraphs. The project proposal is just a few paragraphs. Publications. Gregory, please clarify, what do you mean by publications? Well, at the end, I will give some reference publications for you to start reading on at the end of this session, if that's what you meant. All right, so let's review the schedule a little bit so you have clarity on what each section will be dedicated to. So first of all, we will talk about if results become publishable. There's no agreement. So whatever research you do, it's yours. Our goal is just to support you in what you do for the duration of this program. Okay, so to study different types of communities and their composition, we will talk about sequencing methods and approaches to generate data so that we understand how to generate the right kind of analysis and how to develop an understanding of what that analysis means. Akil, if you haven't used any programming tools, You can complete this course, yes, without, you don't need any programming background. And OmicsLogic code will be an optional portion of the program. It is very simple to follow. So even if you don't know any coding, you don't have to do any installation, and you will be able to complete all of this independently or with our help. We will actually review any challenges that you might have in our interactive sessions and help you out with that. The full program is not free, you are correct. The majority of the people in this session today are already participants. If you have not completed registration, Vipsa could potentially send you a link for completing your registration. And thank you, Dr. Sultan Mahadin. I'm very pleased with your comments. Thank you very much. Okay, so in our next session, September 2nd, we will talk about metagenomics. So again, for those of you that are already familiar with metagenomics sequencing, you don't have to join, but this is something that will give us a review. And to prepare for that session, we have this online course, Introduction to Metagenomics. How do you know that you need to take that Introduction to Metagenomics? If you found it challenging, some of the questions that we discussed in the beginning when I was asking you different questions, you know that you probably should review that course. If you felt like you know everything and you understand the differences between these terms and their meaning, you probably don't have to take that course. So then we will talk about how does the microbiome actually function? How do we understand what is the functional impact of a community? And there are different strategies. Using different databases, we can understand taxonomic composition, but also we can talk about what is the impact or association with a particular condition. So we'll talk about on September 4th. Then we'll talk about the variability in composition. What are some of the methods that we have to analyze and look at particular members of those communities that probably have a dominance or a difference between multiple conditions? Okay, for Mohammed, yes, the full course, the full program is not free, you're correct. And Gregory, I'm more of a biochemist, molecular biologist, not a statistician. What background resources do I have for understanding the statistics? So in each one of these sections, in each one of these courses, we do discuss the analytical steps that are needed to take to process data and analyze the data. And in the interactive sessions, for example, this session on September 7th is going to be focused on the statistical analysis and what are the statistical outputs that are meaningful to do biological interpretation. So I think that both of these aspects, the coursework, the practical analysis, and the interactive sessions should give you a good understanding of some of the statistical methods and their meaning. Okay, now Darsh, I will get to your point here in a few minutes as well. So then we'll go through some examples. For example, the American Gut is a project that was started in the beginning of the Human Microbiome Project, and it was a collection from citizens to contribute to a study of microbial diversity in normal, healthy, and self-reported conditions. So we'll actually take a look at that data set and try to understand how do we see some of that variation, how do we interpret it, and how do we find associations. So it's a very practical aspect of understanding microbial diversity in the context of human health. Thank you very much, Gregory. Another part of microbial impact or functionality is actually an indirect one. So it is not typically suspected that microbiome itself has an association with other types of, for example, neurological type function. So one is a direct one. For example, you can think of sensitivity of the gut and ability to process different foods, or even impact on treatment, for example, immunotherapy, those could be fairly clearly and directly explained by looking at microbial composition. However, some conditions like anxiety are fairly difficult to link to microbial diversity. And so we will take a look at one of these projects that does show how we can separate diet and other types of confounding factors from the direct impact of the microbiome, and look at a project example where we can find such associations and test them experimentally, of course, but we will use a published study to look at that kind of a connection and speak about the gut-brain axis in general, which I think is a very fascinating kind of a research direction that we can explore, and potentially you could further on study in your independent project. So then I think when you think about your independent project, it is very important to understand what types of data sets are available, what types of data should you be looking for, but also how to structure your question in such a way that you can ask a question and know what you will get as a result of your analysis, so that you can make a clear and effective annotation and explanation of the results. So we will actually take a look at how to identify a significant and impactful question, how to then find the right data set, how to apply the right type of analysis and produce the right type of visualization that clearly explains both what you did and the results from a scientific perspective. What do they actually mean? So we'll have a discussion about that, and hopefully by that time you will have some examples. So as I mentioned, September 14th, you will already have submitted some project proposals that we can look at in detail and even practice some presentation of what might be the result of your analysis. Then you will have another couple of weeks to get some feedback on your project. So the goal is not just to prepare a project proposal, but to be able to conduct the analysis yourself, present your research results, and then get some feedback both from the program participants. And by the way, you can work with other program participants together. You don't have to work on your own. You always will find that there's a mix of people who are more experienced on the data analysis side, more experienced on the biological interpretation side, or maybe better presenters than yourself. And so you can always think of these projects as a collaborative effort, and your goal is to get some expert feedback. So potentially, if your results are then publishable and you feel like you have the time and the dedication to take it to that level, we also offer research support after that through our research fellowship program at the end of the program. Let me show you a few practical examples and talk about what are we going to cover as we discuss these project examples. So when we talk about the American Gut Project, we're talking about finding associations between self-reported and some general aspects of microbiome composition based on a microbial sample. So here's a result from the American Gut Sample, for example, where you can look at the different types of bacteria present in your sample compared to some averages by different types. You can see some of the most abundant microbes, and we will learn about how to conduct such analysis. How do we take one participant and see them in the context of everybody else, or compare groups and based on what types of so-called phenotypes that are self-reported. So we can actually look at that project right here, humanfoodproject.com, American Gut. As I mentioned, we will also take a look at projects that have some indirect associations between the microbial community, for example, the gut or the fecal microbiome, and general state. For example, anxiety is a psychological condition, so what might be the relationship between the two? And so we'll see how the specific experimental design and different types of data that are available in the project as a whole can help us study that kind of microbial variation. Now, when we talk about microbes and metagenomics as well, you will see that the term metagenomics also is used to talk about sequencing of genomes or of particular representatives of this microbial community. So we'll also look at how we can apply metagenomic sequences to study such examples of tuberculosis, which is a mycobacterium, Ebola, which is a virus, or rhizobacteria, which is an important type or a class of microorganisms that can have a huge impact on crop production. Other types of exciting research opportunities using microbial data and using metagenomics include the study of treatment impact. So how effective is treatment? For example, when we talk about different types of probiotics, when we talk about different types of treatments that are going through the system, for example, through the gut, the absorption of those elements is also impacted on through the microbiome, and how treatment with antibiotics can actually lead to deregulation of the microbiome communities and have a long-term significance for different types of risks. It is important that these types of studies have actually led to very practical results already. For example, you can see here an article that was published on November 5th in 2019 that talks about the Chinese government approving Alzheimer's treatment that targets the microbiome. So we can actually see very practical outcomes that have come from such research. You can also see that kind of in the future of microbial research, this type of study can have a significance on treatment selection. And today we look at treatment selection that is treatment of clinical conditions as well as preclinical conditions. So different types of sensitivities and different types of diets that contribute to overall health benefits. Ideally, in the context of this program, you will see how different types of omics data could be analyzed using bioinformatics analysis pipelines and reference databases to understand the relationships between clinical and omics data for applications of diagnostic assays and selection of therapeutics, or even an understanding of some basic questions about the impact of microbiome on the human health. So about your independent projects. For independent projects, we have, and for interaction, and one second, I will get to the questions here in a second. For the interaction between all of you, between yourselves and our team, which will support you, we have a forum. So again, if we go back to this page, please take a moment to go to the forum link. So I'll just paste this link in the chat. You will see here already several things that have been posted. Welcome to the forum, session recordings. This is where you will find all of the recordings from these types of sessions like we're having today. Questions about the educational courses. So if you have any questions about the metagenomics coursework that we are going through, you can post them here. Questions about the pipelines. So the pipelines that are not going as fast as you think they should be, or you're getting some errors or not getting the right kinds of outputs, you can post them right here. When you post those questions or those comments, please add the link to your pipeline. What is the link to your pipeline? If I go into this pipeline, you can see that up on top here, I have a link. So this is a URL that has a pipeline ID, and using that pipeline ID, it's very easy to understand what is the challenge that you're facing using our backend administrative panel, where all of the script outputs and inputs are recorded. Also on the forum, let's go back to the forum here, you will see additional resources for the program. And so this is something that you can take a look at today. I pasted here important publications to get us started. For example, Bioinformatics for the Human Microbiome Project, Human Microbiome Project Overview, and some basic concepts for microbiome research, an example from Brazil, a microbiome project from there. Finally, you will see a project submission form. So in the project submission form, you will see a form that is dynamic. In other words, it updates every time that you change something. And this is directly related to some of your questions before. How long is that project proposal? So this is your project proposal. You will see all of your information from the form that hopefully you will fill out today, right here. And then you will also start filling out these project sections. So the project sections include project title, problem statement, or what are you trying to solve, project summary, and this could be several paragraphs long, link to the raw data, so what kind of data have you found for this, and then some reference publications. Do you get a protocol that guides you to create a pipeline, perform analysis step by step? Yes, you do. And most of that will be found inside the coursework, but we will also review that in our interactive sessions. For Gregory's question, multi-omics, will you be learning how to integrate across omics? Not specifically in this case, but we do have a whole section on multi-omics integration, and we will touch on association studies. Those association studies will include genome-wide association studies, association between using statistical association of feature-to-feature correlation, as well as some network analysis that could be done for that kind of a study. I think it will be more informative to take a look at multi-omics integration in the context of a particular project example. So if you are interested in multi-omics integration, I recommend finding a project on a publicly available resource, for example, the NCBI database that has that kind of data. Okay, so again, the objective of this particular section is to submit a project proposal. And to help you in that process, we have a whole article that talks about different examples. Let me just give you the right link here. So we have here a series of recommendations. Sorry. This is the right link. Where you can explore some examples and some guidelines on how to prepare an impactful and interesting project proposal. Finally, our objective is to really help you transition from what you learn in this program to a real research using bioinformatics tools and methods. And we have quite a lot of experience with previous participants of our programs. For example, you can see Olga Udartsova here from Roswell Park Comprehensive Cancer Center that was a participant of an OmicsLogic program before and was able to understand how bioinformatics can be applied to her cancer research, as well as to actually use some of the tools in her preclinical studies and apply even to cancer patient treatment. So I think that's an important outcome that we are very dedicated to in this program, and we hope that most of you are able to actually get this kind of a benefit from this program of not only learning about bioinformatics, but also applying that to your research study. And I think we have the support as well as the tools that will help you make that transition. Obviously, we are not experts in everything, and so we have a panel of external support of mentors that will be evaluating projects at the end. Most of them come from our partners at the Tauber Bioinformatics Research Center. This is a research center that is located in Israel at University of Haifa in Israel. And they have a number of PhD students and postdoc students that are doing research using bioinformatics tools and participate in large consortia projects using bioinformatics. And so we invite them to provide feedback for some of the research projects. That will include some questions and clarifications that you will have to make, but also for the top projects that we jointly recognize as the most advanced projects, they will provide some detailed feedback and an evaluation score. for your project. So hopefully you can use that evaluation score as evidence for the quality and significance of the project that you are planning. We recognize that the projects themselves can take quite a long time to complete, especially if they're real research questions. Nothing goes as planned in any kind of research, including wet lab research or bioinformatics research. Playing around with the data and reanalyzing the data and making your presentations multiple times is something that should be expected. And I'm sure that most of you are familiar with that process, and some of it might be frustrating, but really it is a beneficial process for real impactful and reproducible results. So to summarize, we have 6 interactive sessions that are going to be focused on some theoretical aspects and practical aspects of metagenomic research. During these sessions, we will discuss and review specific questions, data sets, project examples, and interpretation approaches for microbial data. After that, you will have some time to work on an independent project. So for those of you that have already registered, we will have sessions on these dates. It will always be between 10 a.m. and 11:30 a.m. CST, Central Standard Time. During these sessions, we will answer any questions and also provide you additional information at the end of that and post some responses on any questions that you might have. Does the access to OmicsLogic code expire along with this course? Can I utilize it in the future also? Yes, you can definitely utilize it. There's a separate license that you can purchase after the program at a discounted price and use our online server for R. Python code actually loads into your browser session, so you can actually utilize that on your own computer. The Zoom link, yes, at the same time, I believe it is the same Zoom link. Vipsa can confirm that, but you will actually receive an email invitation, maybe 24 hours before each session, that you can confirm and add it to your calendar. So the objective of this program is to understand the microbiome, to understand metagenomic sequencing, to understand methods of metagenomic sequence analysis, and apply that to interpret the microbiome. So that will include some methods of analysis, visualization, and interpretation. Okay, so how can you register? Bipsa, if you are here, please reply to some of these technical questions. Yes, you can use any kind of microbial samples. So let me get to our next. Okay, so for the questions. Okay, and Bipsa, Gregory also had a question about whether the sessions will have the same Zoom link or will it be a different Zoom link. I believe it will be different after this session because this session was a free session for anyone to join and learn about the program also. But later on, we will actually send links. Yes. Okay. Thank you, Bipsa. Okay. Any other questions about what we reviewed up until now? Okay, so in general, I just wanted to finish off with a couple of additional points, and those are related to some of the materials that you can start learning from for our next session. These are the publications that it would be beneficial to review. Bioinformatics for the Human Microbiome Project. So here you will find information on the Human Microbiome Project and what types of bioinformatics methods are applied to what kinds of different types of data sets. Also, there is a link to the NIH Human Microbiome Project itself. This is a data set that a lot of people will use in different examples of how tools work. And this is good for benchmarking and different types of comparisons. And also it is good to do some questions, right? So how can I have some questions that can be answered with this type of analysis? And sorry that that is behind there, but there's also another paper, Bioinformatics for Microbiome Research, Concept, Strategies, and Advances, which is actually a whole chapter from a book. So these are some of the links. Okay, will today's webinar be available because my audio was garbled in the beginning? Yes, the webinars will be recorded, or they are recorded, and they will be available on our YouTube channel. But also you should have a link that you should get an email about with this particular recording after this session is over. Can I store my metagenomics data free of cost at NCBI? Yes, NCBI does not charge for storing the data. If you have a data to submit, a data set to submit, you can go to NCBI and go to submit right here. So the chat links are available right now. Where are these links available? Yes, they are available on the forum. So if you go to the forum, as I mentioned, there's a few different categories for posts. That post was specifically for project submission, but here's a section, additional resources for the program. So if you go here, this is it. These are the links that I just gave you. Okay, so it's great to have all of you here. Thank you for joining today. Just in conclusion, I want to mention that we do have a LinkedIn group that we invite you to join, as well as the Facebook group. Here, I'll just paste the links for those as well. We want to build a community of those that are learning about bioinformatics and are going to apply bioinformatics in their research. We feel that we can exchange information and connect you to other experts as well. We have over 500 people in our LinkedIn group, some of them with some advanced degrees and both background in academia, research, and industry. So I think it would be beneficial to link up with those. And also on Facebook as well, for those of you that rely on that platform, it's called Learning Biomedical Informatics. Okay, Mikhail, it's also great to see you here again. Prerna, can you give an example of a project data where we can understand how to initiate the project? Yes, I feel that some of these example links that I gave are exactly that. So you will see in our, sorry, lost the chat box. You will see on our, in the courses, we actually take examples from some of these practical data sets. So if you have enrolled in Metagenomics 1 or Metagenomics 2, you will see two different examples explained there. But also some of the links to the publications should give you an idea of what kinds of questions you could be asking from these types of data sets. And, okay, so how to initiate the project, actually, that's a great question. I feel like I also touched on that topic, but maybe it wasn't clear enough. This is an overview. So if you go to that link, you will see some outline of what you can do and how to find a topic and how to do some literature research, how to develop your project. So there's, I think, a good overview right here on all aspects, including some examples from previous participants that you will find here at the end. All right, great. Well, thank you again all for joining today. If you have any additional questions, I recommend posting on the forum. And for those of you that are joining not the first time, for those of you that have joined our programs before, it's great to see all of you back. If you are interested in starting to work on your project and you need some support from us, we'll be excited to work with you on developing those projects and making sure that you have all the necessary tools to do that. And I am looking forward to seeing you again on our next session. So look out for those emails. Please try to complete the Introduction to Bioinformatics course for our next session. In that course, there's a survey that we're looking to get to know you a little bit better, your background and your interests, and what are your objectives from this program. So we'll be looking forward to that information. And we're also going to be excited about seeing all of you join our future sessions. We'll talk about next generation sequencing, some approaches, and what you need to understand to make sure that the analysis that you're planning is impactful. So again, we'll see you again next time. Take care.